today. They just weren't able to. But I just want to say that, they've done a magnificent job in terms of really like helping take some leadership in this group. And I just really want to highlight their contributions as well as everyone else's. So this is our working group, and it's morphed a bit over time. So I do want to spend just a moment talking about the history of our working group, because we had kind of one direction that we were moving in when we wrote the proposal, but then had to pivot. And I think that it's important to talk a little bit about. So we originally had a big focus on trying to bring together this emerging field of macrophenology with traditional ecological knowledge, and we had a really thoughtfully, co-produced proposal with many Indigenous members in it. But when we came to have our first meeting, for various reasons, the just circumstances, we didn't have a critical mass from folks who had a really strong grounding in traditional ecological knowledge, in terms of the critical mass that we wanted during the start of the working group. And so we actually had a fairly tough meeting our first time, where we had to say, kind of come to the conclusion that we weren't able to do that kind of co-production and lean into a little bit more of the science questions that we were able to address with the members who were able to come to the in-person meetings. And so I just wanted to recognize that, because sometimes even with the best laid intentions and plans and so forth, you just have to pivot. And I think that was a big lesson that we learned in our group, and I'd be happy to talk about that more later. But here's our group. And, we also had some other challenges, where we had like federal scientists who had to leave the group partway through because their duties changed and so forth. So it was a tough time, but we persevered and had some fun and did some science. And so what our group is really interested in is phenology, which are the biological life cycle events of plants that are recurring through time, and that really focused on the timing of those events. And some of those events can include things like budding, flowering, the emergence of leaves, the emergence of fruits, and then the senescence of fruits or leaves as well. And as you all know, or probably have heard about, that the timing of these events is really changing through time. And this is just some nice visual eye candy from the National Phenology Network, showing some analyses they've been doing with spring indices of blooming, and just showing how the timing of spring, as indicated by plants flowering, or is changing across the United States. And so why should we even care about this in the first place? Well, many of you, if you're like me, are burdened by seasonal allergies. You may have pollen allergies that drive you crazy. There's a lot of our food production is linked to pollination from that same pollen that can cause allergies. Is very important for pollination and fertilization of plants to produce fruits that we eat. There's also all of the interactions that the plants have with the pollinators themselves. And there have been many studies that show that if you have kind of these seasonal mismatches between the activity of the pollinators and the plants themselves, that you can end up with lower yields of fruits. And then also phenology, especially the timing of leaf on and leaf off, is really integrally tied to ecosystem level processes and many of the different kind of nutrient cycles that we see that are extremely important for global vegetation dynamics. So phenology is really important and something that we should all care about on many different levels. And yet we know some about phenological responses to climate change. We know that there can be impacts from variation in temperature and precipitation, but there's still a really black box around how those impacts of changing and dynamic environments can impact phenological responses when you're looking across scales of space and time. And that kind of component of space and time is really what's a large part of macro systems level thinking. Thinking across these larger temporal and spatial scales and those interaction between scales. And part of why it's so challenging to study phenology in terms of through a macrophenology lens is that you really start to have to pull together different data types that have different idiosyncrasies. So we're fortunate in that we have a lot of different sources of phenological data. We have herbarium records, like pressed plants where we can go and see whether or not the plant was flowering at a given time. We have observatory networks, like the National Ecological Observatory Network that's headquartered right with you in Boulder, where they go out and send people to take observations. There's community science networks like the National Phenology Network, where you have school children who can go out and record information about flowering or leaf on or leaf off in their schoolyards. There's app-based records like iNaturalist. And there's also remote sensing data that can either be from a satellite remote sensing or from kind of airborne, more near surface remote sensing. And one of the challenges, though, of pulling all of these different types of data together is that they're measured at different scales, right? So there's different spatial and temporal scales at which all of these observations are measured at, as well as different levels of biological organization and varying levels of taxonomic coverage represented by these different data sources. And Liz Amador first authored a really great paper in New Phytologist, where this figure comes from, where she laid out some of the differences in these different data types. I'm just going to walk you through them to kind of orient you a little bit more. So in terms of biological organization, when you have herbarium data or app data, community science data, or observatory networks like NEON, you're oftentimes looking at the scale of an individual or a group of individuals. Whereas oftentimes when you're looking at remotely sensed data, you're getting an image that's just kind of an overview of a community. So there's a difference there in biological organization and resolution. There's also differences in the taxonomic or phylogenetic coverage of these different types of data sources. So herbarium data and apps tend to be very rich. They're phylogenetically and taxonomically with total coverage based on an estimate that we did when we wrote this paper was over 5,000 total species. Community science, are then the next highest in terms of taxonomic coverage. Data from NEON have around 150 species, but then when you get to remote sensing, oftentimes because you're at a coarser resolution in terms of biological organization, you're also looking more at dominant plant functional types rather than species. But that is changing. So with some of the hyperspectral data that you can get from remote sensing, you can use neural networks to identify crowns of trees to species, and then you can start to kind of dig into that taxonomic resolution a little bit better. But overall, people historically have kind of looked at dominant plant functional types from the remote sensing data. And then in terms of spatial resolution, with herbarium data and apps, you oftentimes have just that single individual that you're looking at a point in space. With community science initiatives, you're oftentimes looking at multiple individuals per site. And with NEON, you kind of oftentimes have a slightly larger area that you're covering with a 800 meter square plot on average with the phenology loops that the field crews run along. But once again, when you're at that kind of remote sensing level, you're thinking more about, how many plant functional types you're seeing per pixel, and so that's going to be really dependent on the pixel size of the image. ROI here would be the region of interest if you're using one of Andrew Richardson's phenocams, those like old school security cameras that they point out into places to get the phenology data. And then the temporal resolution is also variable. So your herbarium and apps are very opportunistic. It's just kind of when people go out. Oftentimes it might be at the peak of the blooming season because that's when it's most obvious to go out and sample. Um, with community science and NEON data, since NEON follows the same National Phenology Network sampling protocol as the community science, you oftentimes get more regular kind of weekly intervals of revisits to plants. And then with remote sensing, it can be highly variable. It can, if you're using a satellite remote sensing platform, it may be every 2 weeks, depending on how often that satellite flies over the Earth. But we're getting much higher temporal resolution with some of the newer missions from NASA, for instance, like the PACE satellite, which was actually for ocean observing, observes the terrestrial areas as well, and you can get daily coverage there or bi-daily coverage. Um, and in terms of temporal extent, herbaria are really where it's at if you want to get a deeper time series, because we have data that go back in time. All of these other methodologies are really looking more, since like the 1980s or early 2000s. And so, a big challenge for macrophenology is you want to cover all of these ranges of space and time and levels of biological organization, but you have all of these data that are in different formats, coming with kind of different biases to how they've been observed that you need to take into consideration. And so this was just a really nice kind of preliminary look that Liz and some of our other working group members took to kind of see what was available. But the key thing I'm trying to get across here is just that plant phenology is really messy. And a big goal of our working group was to try and understand how phenological responses of flowering plants in particular across the contiguous United States vary using these kind of open and multi-sourced different types of data. And one of the key questions that we've been focusing on is, do species change their location when they're presented with differences in the environment, or do they change their timing, or maybe both? And so for the study that I'm going to talk about today, we focus primarily on herbarium records, observatory network data, and community science network data. And that's kind of the key data sources that we're considering. And that's to understand changes in peak flowering time. But then we also wanted to have this kind of understanding of whether or not species were shifting their geographic distributions in addition to, or not in addition to, shifting their phenological response of flowering. And so for that, we used some other different types of data. So we did use the herbarium data again, but mainly just to look for species occurrences rather than flowering. We used observatory networks like NEON and the USDA plants and the United States Forest Service. And we used app-based data for that from IDIG Bio and MBN. And then we also use community science networks like the National Phenology Network. So for this, we use slightly more different types of data for pulling together information on species distributions. And again, the focus here in this study is on flowering in terms of the phenophase of interest. So just to kind of lay out the premise for our study, we were interested in how flowering could shift with climate change, but also how there might also be concomitant geographic distribution shifts of the plants themselves. And then, we wondered if there might be some sort of trade-offs under climate change or any kind of interactions that we might see. And to motivate our hypotheses a little bit more, here's some conceptual diagrams. So you can think about how a plant might need to, if there's not a lot happening kind of in terms of environmental dynamics, there may not be a need to change the range the geographic range of the species. So the geographic change in geographic range is on the x-axis, and then change in phenology is on the y-axis. And we're trying to, when writing our proposal, we were thinking a lot about how through a macrophenological lens, you could start to look at kind of differences in the, in low latitude and high latitude species. You could look at differences in native and invasive species. And so we framed our hypotheses along those lines. And one of the things that we considered is that there could be a latitudinal constraint or trade-off that we might see, where potentially high latitude and low latitude species might differ in their responses. So high latitude species in the temperate zone might have just over time seen more climatic heterogeneity at range edges. Whereas at low latitudes, maybe there's more of a consistent climate. And so we expected that, if presented with changes in the environment, higher latitude species might just be more capable of changing their geographic range, but also changing their phenology just because they've experienced more different types of climate heterogeneity by being in these higher latitudes. In contrast, the null hypothesis would be that we maybe don't see that kind of interaction term. And then a second hypothesis that we had was that there could be differences also depending on the origin of the species that we're looking at. And so perhaps we might see a species origin constraint where we might see differences in responses in terms of this interaction plot for invasive and native species, where invasive species may be more likely to change their range and their phenology if presented with environmental change, because they're more plastic in their responses generally, and also because they tend to have more dispersal capacity. And then our null hypothesis would be that we wouldn't see that kind of interaction plot. So these were the two hypotheses that we wanted to test. And the way that we went about this was through a species distribution modeling framework, where we wanted to model changes in phenology and also changes in range. And when I say range, I mean geographic range. And so in order to calculate that change, we needed to be able to run species-specific models at two specific time periods, so a historical period and a more contemporary period, and then calculate the difference between those two time periods. And then in our workflow, we eventually take those SDM outputs and put them into a Bayesian regression to test those interaction terms. And so these are what our data look like. So in order to model phenology, we wanted to take our herbarium records, our observatory network data, and our community science networks. And we wanted to be able to model just as you would with a species distribution model, where you're modeling occurrence, presence or absence of a species. We wanted to model presence or absence of a flower. And to do that, you first have to define what you're going to determine as the presence for a flower. And so for each of the neon domains, we looked at the herbarium records, observatory networks, and the community science network data. And we identified with quantiles the peak flowering time window for each of the species in each domain. And then we were able to say whether or not an observation fell in that peak flowering time period and also had a flower. And so we had this point level data that we then gridded up, and we could assign to a given pixel in a raster whether or not there was a presence or absence of flowering in the historical or the current time period. And so this was a scenario where we did have presence and absence data for this kind of it's a species distribution modeling framework, but we're modeling phenology as a response. And then for our for actual species distribution models, we're modeling the geographic range, the occurrence of a species. We had presence-only data, where we had the herbarium records, observatory networks, the community science network, and the app-based records. And those were all only presence data. And so we were able to use a species distribution modeling framework where we had presences generated pseudo absences, and then we're able to model the occurrences of those species. So when I was referring to those current and historic time periods, the time periods that we identified were for current were 1985 to 2025, and for historic were 1910 to 1950. And that was really a decision that we made based on previous studies folks in our group had done, and then also just looking at kind of the distribution of information that we had coming from the herbarium data and the more current observatory network and community science network data for phenology, so that we could kind of have even sample sizes and make sure that we had good coverage of species and so forth. And also there's a good argument to be made that, you know, post mid 1980s, climate change starts to really ramp up. And so we are in kind of like a different biologically relevant period as well. In terms of species, once we did all of our data cleaning and so forth, we ended up with 144 different species in this data set. 54 were woody and 92 were non-woody. And so as you'll see in models that we run later, we did take into account differences in functional type, just in case there were differences, given that woody, non-woody split in the data. When we test the species origin constraint hypothesis, we only use the non-woody species because there just wasn't enough of a balance between woody and non-woody when you start looking at invasive and non-invasive. And we were trying to have pairs between genera to make everything balanced. And so there's less data in our invasive species kind of origin constraint model that we looked at. As I mentioned, there's this kind of, you know, it's important to point out that our responses are slightly different for our phenology and our species occurrence data sets, where we did have kind of defined presence and absence for flowering, but really, you know, presence only data going into the occurrence modeling for the geographic distribution model. Just it's an important caveat to consider. And the climate covariates that we chose through model selection for our analysis were temperature as represented by spring anomalies and annual averages, and also precipitation, also represented by spring anomalies and annual averages. And those came from the PRISM data. Okay, so as I mentioned, our species distribution models, you know, for both flowering and geographic distribution were species specific. So for the peak flowering, we did an ensemble modeling at first, but all of the models, both for flowering and geographic distribution, were best represented with random forests. So we just went with that. So we went through and we fit a time 1 and a time 2 model for phenology, and then we took the difference between those to get the change in phenology. And we did the same with our occurrence data. And then what we did is apologies for all these equations here, but I think it's something that's really cool that we actually did in this study, is that we wanted to propagate the uncertainty from these models. And so for every single pixel, you know, we ran these models 10 different times. And for every single pixel, we, you know, from the random forest, calculated the standard error essentially from it. And we're able to kind of propagate that uncertainty through when we ran our Bayesian model to test our species origin and our latitude constraint hypotheses. And so I think that's something that I haven't seen done all that much in the literature when folks are using these kinds of models for this kind of question. And it actually took a lot of time and a lot of compute on Cyber. So I just wanted to really highlight that because we took a lot of time to do the error propagation. All right, so then we ran all of these distribution models, all of these phenology models, and then we wanted to actually take the outputs of our models and see if we saw these hypothetical graphs that we had shown you before. And so we put these into a regression model in a Bayesian framework so that we could propagate all that uncertainty through. And this is just a written out version of what the model looked at like. But what we did is we used that probability of change in peak flowering as the response on the left-hand side of the tilde. And then the probability of change in occurrence, that difference in ranges, was actually a predictor variable of that change in peak flowering, just as it's on the x-axis in this diagram. We then accounted for latitude, functional type, because we had those differences between woody and non-woody species that we wanted to just make sure weren't driving any of the trends. We really wanted to test for that latitude difference. Sorry, ignore the range difference times status. That should be in the next one. I made a mistake when I put together my slide. Just ignore that term. And then we had a random effect for species. And we also accounted for spatial autocorrelation through a spatial term, which was a conditional autoregressive term. And there's a fun thing you can do when you're working with neon data is that, if you have if you're trying to account for spatial autocorrelation in a model and you are looking at all of your different points, it becomes this like really intractable mathematical computational problem because you have to calculate the distances between all your points. But a special trick that you can do when you're working with the neon data or anything with like an ecoregion or anything, but with neon we have the domains, is that you can create an adjacency matrix where you have your for any given domain, you look at just the adjacent domains to it to calculate the spatial autocorrelation term. And that makes it a much more computationally tractable problem. And so that's what we did for this one. And so here are our results. I'm just showing, once again, we ran this 10 different times for all the error propagation and so forth. And actually we also ran it 10 different times because we started to hit up against, even with all of the tricks that we were using to reduce the computational load, there's just so much data. But all of the runs that we did that were randomly pulled from space all had similar results. So we're just showing you an example from run number 8. And what these are the coefficient plots from the Bayesian model. So you're seeing credible intervals here. It's the credible interval overlaps the dashed line of 0. That means that it's not a statistically significant effect. And so all of those instances where you have that overlap with 0 by the credible interval are shown in gray. And then significant effects are shown in blue. And we also included dispersal in our models to see if there were differences in different dispersal types too, because we had some interest in that. But what you can see is that the main pieces that were important were that interaction term between the change in range and latitude. Latitude itself was only kind of marginally significant, and then our spatial effect was very significant. We see in this interaction plot really kind of, these are latitudinal bands. I don't show all the points behind here because it's just way messy. But there's the latitudinal bands, and you can see that we really see that interaction that we expected, which is pretty cool. Where our high latitude plants are showing much more change in phenology and change in range relative to the low latitude when there's a need for a shift. And this is the results from the functional type model where we did have that functional type term in it in the model, but that functional type, once again, was not significant. So we didn't see a really big difference between woody and non-woody species. Then we also looked at this for sorry, I put these slides together last minute. Nativity and status just mean whether or not it's invasive. Apologies for that. But we ran essentially the same kind of model, but we looked at whether or not a species was invasive. And we didn't see any suggestion that nativity status, whether or not a species was invasive, had any sort of impact. So we didn't see support for that hypothesis, but that kind of change in range by latitude effect is still there, and the spatial effect is huge still. And so you can see that in that plot there. So, overall, we were interested in this idea of, are there trade-offs between phenology and changes in phenology and changes in the distributions of species in terms of their geographic range? Overall, we can see that climate change is reshaping these kind of plant phenological trade-offs in ways that reflect variation between species and their geography. But we didn't really see strong evidence for, big differences between species origin in terms of if it was a native or non-native species, or the types of dispersal modes or functional types of the organisms when we look at it at this macro scale. But we did find, strong support for a trade-off between phenological shifts and geographic range shifts that are mediated by latitude. And, I do wonder a lot, right, if latitude is really picking up perhaps, photosynthetically active radiation and so forth, because in a lot of ways, latitude is very correlated with PAR. And PAR is so important for species when we're thinking, for plants, especially in terms of when we're thinking about when they have time kind of metabolically to rest and when they have time to be active. And so it's just, I think that this study would also benefit from, much more of a physiological lens as well. So at the end of the day, I hope that we've been able to take this crazy ball of string that is all of the phenological data options that we have out there, and add a little bit of structure to it and try to make some sense of it. When you're working at these larger scales of space and time, things get pretty messy, and so it's trying to figure out those patterns in there. And I do want to just say that this is just one of the papers that our group has been working on. We have another paper that's still cooking, and that will be presented by Hanshi Chen. And Hanshi is a PhD candidate Hanshi just passed her qualifying exams, so good job, Hanshi. And she's at Duke University working with Tong Chu. And Hanshi's been working on some of the phase 2 neon phenology data where they have, it's essentially the NPN, the National Phenology Network style protocol that they use on the phenology loop at the neon sites. But for the first 5 years or the first, many years of neon, they focused essentially on just a few dominant species where they got that really rich field observations on the ground of flowering and leaf on and off. But in phase 2 of the phenology protocol, they reduced the number of individuals of the dominant species being studied, but increased the species coverage. So you can actually start to look at community dynamics, which is pretty cool. But what Hanshi's looking at is, within species, are there differences in green up and senescence rates using those phase 2 phenological data? And then I'm going to present a shortened version of this talk at the organized oral session for ESIIL. So if you haven't got enough of it, feel free to come and see it again, or we can talk more there. And then I also just want to advertise a couple other things. I'm separately from the macrophenology group. I'm part of a group called Imagomics, which does a lot of computer vision, um, and and they're creating over the course of the last 5 years. They've created as a Harnessing the Data Revolution Institute, funded by the National Science Foundation. The Imagomics Institute has created a lot of really cool tools for computer vision for biologists, and we'll have a booth there in the conference center for the whole meeting. So feel free to come by and learn about some of those tools. If any of you are involved in the Sentinel Beetles hackathon that ESIIL held last winter. Um, that that hackathon was put on by Imagomics, and so you could stop by and chat with them and maybe see some familiar faces. Um, and then one more plug for ESA. My A graduate student I collaborate with, Allison East, is really involved in the early career section for the Ecological Society of America. And they're currently seeking both next generation scientists who would like to be mentored, as well as mentors who would like to self-nominate for pairings during the week of ESA in Salt Lake City, just to have, I think there'll be like a lunch session, and then you can kind of have times where you meet up with your mentor and just chat about different career options or strategies. So I think they're hoping to get applications on by Friday. So if you want to just go ahead and take a picture of that QR code, I'll leave it here for a second. All right. And then I just want to, thank some folks. We had a lot of helpful comments from some colleagues that we've got listed here. Neon staff are always phenomenal, right? So we were super lucky to have two Neon staff members in our working group, Katie Jones and Eric Sokol, who have been fantastic contributors and collaborators. We also had some really great help from Claire Lunch and Chris Florian. So Claire has been doing some really cool work with some of the other Neon staff to essentially upgrade Neon utilities, which is the R package used by Neon. And so you can actually use cloud native formats with Apache Arrow so that you don't have to download all of those zip files anymore to actually access tons of data and query them. So she showed us how to use that, and we were like totally blown away. And it's getting to be really important as the Neon data repository grows. And then Chris Florian gave us some really good tips. We were trying to kind of use some of the terrestrial instrument sensor data, like temperature and precipitation data from the tower for our analysis. And we had a lot of questions around that. We didn't actually end up using it because there were some hurdles if you guys want to talk about it. But he showed us a lot of kind of ins and outs with that data set that were extremely helpful. And I'm sure I'm forgetting someone from Neon, so I just want to thank Neon. And then all the ESIIL staff, and I didn't even try to name names here because I knew I would like leave someone out and I didn't want to be exclusive, but ESIIL's been just so welcoming, so helpful. We really appreciate the work that you're doing. It's incredible. And then just some additional support that we've had from NSF that was funding Liz Ometer for her work as a graduate student. So thanks everybody, and I'd be happy to answer any questions. And I haven't been monitoring the chat, so I'll probably stop sharing my slides so I can see the chat too. Thank you.
Sydney, it was very notable how significant that spatial effect was in your Bayesian model.
It always is.
I know. How do I interpret that ecologically? Is this neighbors preventing movement into a new area, or how do I interpret that?
No, I mean, I think it's just that, spatial autocorrelation, is one of the first principles of geography, is just that points that are nearby each other, whether in space or time, are going to be more similar than points that are further away. And that is just pervasive, right? And I think the key thing is that not that that was the biggest effect, but that after we accounted for that, we were still able to, account for some of our other factors of interest. And, a lot of studies just don't account for that. And then all of that variation that would get sopped up by a spatial or temporal autocorrelation term gets pulled into other variables if you don't account for it. So that's what I would be more worried about than it accounting for a lot in when we do account for it.
Cool. And then that increased plasticity in the high elevation where they're able to change phenology and range more. How do you think that'll play out? Like, what does that look like if we start to think about conservation in the future?
Yeah, so it's not elevation in this study, it's latitude. Because if it were just in an elevation, you're not in a good place, right? Because there's only so far you can move, right? Um, uphill. But the elevation piece is interesting because a lot of our phenological observations are not really well filled out in along elevation gradients, surprisingly. I'm doing some work in a separate project with Chuck Davis and some of the folks from the Rocky Mountain Biological Lab to pair herbarium data in alpine areas with long-term phenological observations from Rumble. And that's one of the only times where we have those kind of field and herbarium comparisons that we can make. But there's a real lack of kind of good phenological observation in alpine areas. And having worked in the alpine, I think it's because it can be just so hit or miss, like if you're up there, right, when the flowering happens, because you're at that kind of abridged growing season.
Cool. Thanks for all your really cool work.
Thanks, Ty. Thanks for all your help with Cybers.
Any other questions? And if you're online, you're welcome to unmute or add questions to the chat as well.
I had a quick question. Very nice talk. I was wondering, when you're deciding for the Bayesian regression between the response variables and the predictor variables between change in range and change in phenology, is there a situation where you could see flipping those two in like a scenario where, say, plasticity limits the need to change your range, plasticity and phenology, and then would you expect that to affect the outcome, or probably not so much?
Yeah, I mean, I think that's a really great question. So we drew these graphs like every which way that you could. We had a lot of fun on the whiteboards. And we definitely had the axes swapped at one time, and we were, trying to decide. And I mean, it could have impacts. There's a lot to be said with kind of looking at in separate studies, I've looked at kind of how slopes of lines will change and how you can, through those slopes, tease apart differences in plasticity and adaptation. And Kaiju's also done some work thinking about that as well. So it definitely is something that is sensitive and that we should probably, without, overworking the data, probably before submission, take a look at kind of swapping that around a little bit, because it was very much something that we were considering as we had discussions and we're kind of thinking through the thought process. But at the end of the day, we're most interested in that kind of like phenological response. And it gets so tricky with teasing apart plasticity and adaptation in some of these studies. And so I don't think we quite had the right setup for this one for that.
Right, that makes sense. Thank you.
Yeah. Thank you.
Hi, Sydney. This is Jennifer online, if you can hear me.
Hey, Jennifer.
Thanks for such a great talk. I want to ask for a little bit of help. We are about to go into a site visit for our renewal in a couple weeks. And I would love to be able to say, like, what are the new scientific discoveries that ESIIL has precipitated for your working group? Like, I heard you say a couple of things on the data and compute front. But like, how did we help you advance understanding of phenology with big data and big compute?
Yeah, so I mean, I think the big piece here is, as you have all of these different these different data types, a big part of it is just wrapping your head around all of them, right? And so one of the things that we did with the support of ESIIL was really convene, the intention here was to convene a bunch of folks who were already studying phenology using all these different data types so that we could really interrogate them and think about them, pretty stringently and think about the pros and cons and what's possible and what's not with them. And that really informed our modeling approach, right? Because a lot of times, like we didn't feel like we could just kind of pull all these data together and not do all of that propagation of uncertainty, because they're different, right? Herbarium data are collected differently than the observation network data, than the MPM data. And so we felt much more comfortable scientifically in being able to look at model outputs of those data, which is why we did all of those distribution models. And then, but then we needed the compute in order to really be able to do all of that error propagation and thinking about the uncertainty. Because we were, all of those uncertainties were calculated for each individual pixel in these rasters that cover, the whole United States on a 4 kilometer grid. And so it ended up starting to be quite computationally intensive. And, we were really running Cyverse hard. So, I think that had we not had what ESIIL really, the special sauce from ESIIL for this was really kind of bringing everybody together to talk about the ins and outs of the different data types. And then, at what point we would feel comfortable being able to merge them. And that was the decision about the uncertainty propagation. But then that became, when you start doing that across all these pixels and model runs, and then also add in like the spatial autocorrelation, it just starts to really blow up computationally. And so that's where, the resources that any single one of us could bring to the table wouldn't have been enough. And that that summing us all together, we were much more, I'm always bad about that. We're more than the sum of our parts. I can never remember that saying quite exactly, but I think that's where it was really helpful to have the resources from ESIIL.
Yeah, right on. So if I said something like big teams, big data, big compute to advance our understanding of macrophenology across the conterminous US in every pixel that's in every 4 kilometer pixel, like ESIIL enabled you all to tackle questions. And then what?
And the uncertainty piece is the big one, right? Because that's what most people would just ignore.
And when you say addressing the uncertainty, like I'm interpreting that as part of the challenge of the harmonization. So when we're talking about doing data harmonization from museum collections to satellites, like there's uncertainties that get propagated across all of those different sources. And this group, the macrophenology group, has really tackled that problem head on. And now they have a framework and a data set that they can interrogate to ask dozens of questions. Something like that?
Yeah, I think so.
And then are there other pieces, like elements, because phenology is not my area of expertise. Like, are there any new questions or new insights? Or is this the first time that this has been done at this scale? Do you know what I'm getting at? Like, what's the novelty?
Yeah, no, I mean, I think I would say that this is the first time that it's been done at this scale with the uncertainty added in.
Right on.
And I think that's important. Yeah.
Awesome. Thank you.
Yeah. Good luck with the site visit. We'll be cheering for you. And if there's anything we can do to help, don't hesitate to reach out.
Yeah, thank you. Well, if there are, in the future, as your papers come out, if there's these like aha moments, or if you want help on the media front too, we'd be happy to get our institution involved to mirror whatever yours is doing in terms of getting the word out.
Awesome. Cool. Thank you. And just as a plug, there is going to be a hackathon called Florapalooza if anyone's interested in phenology. and images. So Imagomics is going to hold a hackathon called Florapalooza in August. I think the call for participants will come out sometime either this week or next week. But I can send that through the ESIIL channel too. So if people are like, I know many members of our group are interested in kind of being able to automate the extraction of phenological traits from images, whether they be from iNaturalist or herbarium specimens. And Daizhong Li has done an inordinate amount of work doing that with Phenobase. But if people have new images and they want to try working on that, Florapalooza would be a good way to kind of learn some of the tools from the computer vision side of things.
All right. I don't think we have any final questions, right? All right. Well, thank you, Sydney. And
yeah. Thank you so much.